Good morning, good afternoon. First of all, I would like to thank the organizer of Vaccine World Asia Congress to invite me as a speaker to present the development of a needle-free COVID-19 gene-based vaccine. However, before presenting Covigen project, I would like to introduce our company, Bionet. Bionet is a French-Thai fully integrated biotech company focusing on the development and manufacturing of genetically designed vaccines. Today, we are the only manufacturer of recombinant pertussis vaccines. We have 20 years experience in recombinant DNA technology, and we have a global team in Asia, in Europe, in Australia, fully dedicated to develop the next generation vaccines against reemerging and pandemic disease. Bionet was founded in 2001. We started our project on recombinant pertussis in 2006. It took us 10 years to bring this project to licensure of two vaccines, boostergen and pathogen. We have various collaboration with international organizations such as PATH, and also collaboration with universities in Switzerland, but also in Australia, where we have a clinical trial with our pathogen that has started recently. We are also working on the development of various vaccines. One of them is the DNA vaccine against Zika dengue in collaboration with the Institut Pasteur from France. This has helped us to move much faster on the development of a DNA vaccine against COVID-19. And we're going to enter in clinical trial in Australia with the same organization that are testing our pathogen vaccine. As I mentioned earlier, we have an expertise in recombinant vaccines. We have our own patent for pertussis, and we have published several articles on the results of our clinical studies in the Lancet, and some papers refer to our vaccine, papers from WHO or US FDA. Now, let me move on the COVID global situation, and unfortunately, this slide is not updated as the number of cases are increasing every day. We are now more than 4 million cases of COVID in the world. Southeast Asia region is mainly affected, and there are more than 1 million deaths in the world. The Scientific American Journal discuss about how to develop a virus vaccine. And there are two ways. One is a traditional process with inactivated and subunit vaccines. Another way is to use genetic engineering and to bring DNA or RNA vaccines. Another paper from Nature asked the question, can we take a bet on nucleic acid vaccines? And in Thailand, two teams decided to take this bet. One of them from the Professor Kiat from the Chulalongkorn University, who's developing a vaccine, mRNA vaccine, and Bionet developing a DNA vaccine. Both organizations were already collaborating since 2017 on a DNA technology platform. So the nucleic acid vaccine, unlike other vaccines, they are not administrating the antigen, but the genetic information, so that the cell can then produce either the mRNA or the viral protein that will then induce the immune response. So there are two projects in Thailand. One quickly is about the mRNA led by the Chulalongkorn University. This is a global collaboration that involves also the University of Pennsylvania and a different biotech organization in the US. And Bionet will be the industrial partner to produce the vaccine. Now, I would like to focus on this co-vision project that involve also partners in Thailand for studying and testing the vaccine. Now, I would like to start first on the rationale about the S1. So on the left-hand side, you see the picture of the COVID, of the SARS-CoV-2 and the spike, the S protein. On the right-hand side, you see that the S protein is composed of two, and we have selected the S1, which contains the functional receptor binding domain, RBD, that is responsible for binding to the host cell membrane receptor. So the S1 protein is a great epitope candidate that can induce protective immune response, but also minimize the chance of ADE, antibody-dependent enhancements. That is why we selected this S1 protein as a candidate vaccine. We also selected the DNA vaccine technology platform because of the rapid design and development. We're able to cope with a new target antigens. The production time is shorter than with other technologies. It requires a smaller manufacturing facility. The process is reproducible. We can reach a large scale of production. In terms of safety, the DNA vaccine have been evaluated in several clinical trials and have shown a good safety profile, even if there is today no DNA vaccine license for human use. There are for veterinary use only. The production of DNA vaccine do not require BSL-free manufacturing facilities. There is an ease of storage, and the DNA vaccine has been shown to be able to stimulate both humoral and cellular immunity. The target product profile that we wanted to get is a DNA spike S1 vaccine, which can be administered intramuscularly or intradermally. The dose regimen would be a 2-dose schedule with 4 weeks apart interval. There is no adjuvant. The vaccine has to be efficient. The vaccine ideally should be stored at 2 to 8 degrees, and the vaccine will be presented in a vial form with an injector system to transport the DNA into the cell. Let me now present to you the preclinical development that has been done in 3 animal models: mice, guinea pigs, non-human primates. We have tested 2 vaccine candidates. We have tested 3 delivery systems, different dose schedule, dose range, and 2 routes of administration. The readouts are binding antibody by ELISA, neutralizing antibody with a surrogate virus neutralization test, and we have also evaluated T-cell response by ELISpot. The endpoints after the mice studies, we were able to select 1 candidate vaccine and the schedule. With the guinea pigs, we confirmed the effective device and the routes. And now in non-human primate, we have confirmed the safety and immunogenicity at the maximum human dose. Here are the results in guinea pigs. So we gave 2 doses, 4 weeks apart. And you can see that the vaccine was able to induce anti-RBD IgG in both IM and ID routes, We could see that with the 200 microgram ID route, we could induce a higher antibody response than the IM, and increasing the dose in the ID group will not significantly increase the antibody response. So there is certainly a potential for dose sparing with the intradermal route. Let's move now to non-human primates. This is a safety pharmacology study where we evaluated the safety of two doses. So there are 3 doses tested here, 4 weeks apart. And you can see that after the first dose, the vaccine was able to induce anti-RBD response. This is a vaccine injected by Stratis via intramuscular route. And you can see that the vaccine was able to induce a neutralizing antibody response after the second dose. So the cutoff here is 20%, and above this level, the vaccine show neutralizing antibody. We have also evaluated the T-cell response, and you can see that after 1 dose, the vaccine was able to induce a T-cell response specific to S1, as we have also evaluated the S2 T-cell response, and there was no response. So the response is really specific to S1. The response was high after 1 dose and is stabilized after the first dose in non-human primate. In terms of production, we produced the clinical lot in 70 days. But first, let me go back to the key dates. On January 10, the sequence of the SARS-CoV-2 were published. On March 10, WHO declared the COVID-19 a pandemic. We started our project on March 1st with this construction of the DNA with 5 collaborators in the research lab. And then gradually we increase and mobilize resources, both in terms of facilities, but also in terms of personnel. On April 1st, we stopped the production of pertussis vaccine, and we converted the plant into a COVID GMP facility in less than 1 month. We started the GMP cell bank and the preclinical studies on April 20, and we're able to produce the first clinical lot by May 10. When dealing with DNA vaccine, what is important is also how to transport the DNA into the cell. And we have a collaboration with PharmaJet that have 2 different needle-free injector. The first one is a Stratis that administer the vaccine intramuscularly, and we can inject a 0.5 ml. The vaccine, the device is being used with a flu vaccine and is commercialized in the United States. While the second device, Tropis, is to deliver the vaccine intradermally. The dose is 0.1 ml. The vaccine is being used for polio vaccine program for dose sparing with WHO. The device is WHO pre-qualified. Both devices have CE marks. Following the good preclinical data, we're moving to clinical development, and we've been collaborating with Associate Professor Nicholas Wood from the University of Sydney, who will be the lead investigator of this phase 1 trial. This is the first DNA-based COVID vaccine trial in Australia. This is a collaboration with Vax4COVID, which is a partnership of 4 of Australia's most experienced vaccine trial sites. And we're going to enroll 150 volunteers. The organization involved are Teleton Kids from Perth, the University of Adelaide, Scientia, the Government of South Australia, Westmead Institute, and our partner in Australia, Technovalia. So what would be the next step if the results of phase one are found positive? Then we are a biotech company and we have to move on vaccine supplies, and we believe it's possible through collaborative networks. So after the phase one, then we have to consider the phase two and phase three trial, and hopefully we can get an emergency use approval. For this, we need to collaborate with several countries, like even if we're based in Thailand, we're collaborating with Australia with multi-site. So this is the example of a collaboration Facts for COVID. And we have received a grant, or more exactly, our investigators have received a grant from the government of Australia. In parallel, we have to think of the production. We have produced clinical lots, and the next step will be the production scale-up. We are already in discussion through Technovalia with a CMO in Australia, Luina Bio. We are open for technology transfer, and I remind that the DNA vaccine require only the BSL-1 facilities. Of course, this can be done with government funding, like for other companies. Then we can move to the supplies. We have supplied the clinical lots. We have to consider the large-scale supply. And here we aim at supplying vials with the needle-free injector systems from PharmaJet. We can also supply bulk, ready-to-fill or concentrated. And we're also open for licensing like we have with Technovalia in Australia. So this is my last slide. I would like first to thank all my collaborators in Bionet, but also all the collaborators that have been collaborating with Bionet and all the researchers in the world who work relentlessly in the search for a safe and effective vaccine to protect all of us against COVID-19 disease. Thank you very much.